Evening, friends. Professor Brahma Singh Horticulture Foundation, BSHF, a not-for-profit organization, and myself welcome you all to the talk number 25 of this series 3H, Horticulture for Health and Happiness. BSHF is thankful to Syngenta, Patanjali Ayurved Limited, and NHRDF to sponsor this webinar series. I welcome Dr. Pritam Kalia, co-organizer, and Rafi Ahmed Kidwai Awardee, and former head Division of Vegetable Science, ICAR-IARI New Delhi. The talk this evening is on an interesting topic, contributions of soil health to human health through fruit crops by Dr. A.K. Srivastava, Principal Scientist, Soil Science, ICAR-Central Citrus Research Institute, Nagpur, Maharashtra. BSHF is thankful to Dr. Srivastava to honor its request to deliver the webinar. Friends, we all are concerned about human health. Human health, as we know, is dependent on the food one consumes. The food comes directly or indirectly from soil. So, in this chain, soil health is important to contribute to the health of plants, animals, which are the major sources of human food, leading to the state of human health, whether it is a good health or otherwise. Soil has a considerable effect on human health, whether those effects are positive or negative, direct or indirect. Soil is an important source of nutrients in our food supply and medicines such as antibiotics. However, nutrient imbalances and the presence of human pathogens in the soil biological community can cause negative effects on human health. There are also many locations where various chemical compounds are found in soil at toxic levels. The soil of urban environment has received increased attention in the last few years, and they too pose a number of human health questions and challenges. The microbiota of human gut and the plant rhizome are similar in many ways and indirectly connected. Intricately connected, not indirectly, intricately connected with each other. A healthy plant, therefore, affects human microbiota and human health. Healthy soils yield healthy plants for better human health. Perhaps beneficial microbes in the soil, food and human gut are interconnected to contribute to the human health. Today, we are here to enrich our knowledge on the benefits of soil health on human health through fruit crops. Dr. Srivastava, with the rich experience on the subject, is going to talk on this aspect with empirical evidences. Friends, questions, if any, can be raised. They will be answered after this session. Now I request my colleague Dr. Kalia to introduce the speaker of the day, Dr. A.K. Srivastava ji. Dr. Pritam Kalia ji.
Thank you, sir. Good evening, friends. most respected Professor Brahm Singh ji, organizer of this webinar series, my colleague, co-organizer, Dr. Shailendra Rajan ji, and today's most experienced speaker, Dr. A.K. Srivastava, who has had his master's and doctorate from Banaras Hindu University and is presently principal scientist at Citrus Central Citrus Research Institute. Dr. Srivastava extensively pursued his research work on different aspects of citrus nutrition, like nutrient constraint analysis of citrus orchards by developing DRIS based soil plant nutrient diagnostics, orchard efficiency modeling, targeted yield-based site-specific nutrient management, exploiting spatial variability in soil fertility, citrus rhizosphere-specific microbial consortium, and soil carbon loading, INM model, fertigation scheduling, nutrient mapping using geospatial tools, nutrient dynamic studies, transformation of soil microbial biomass nutrients within citrus rhizosphere, and soil fertility map as the decision support tool for fertilizer recommendation. Dr. Srivastava has handled more than 30 projects. Out of 30 projects, 13 as a principal investigator and 17 as a co-principal investigator, exclusively working on fruit nutrition. Dr. Srivastava has evaluated 55 MSc theses and 22 PhD theses and has acted as co-guide for 12 MSc and 5 PhD students. Dr. Srivastava has more than 171 research papers, out of which 102 papers in Indian journals and 69 papers in foreign journals, and 49 policy review papers, which 32 in Indian journals and 17 in foreign journals, 120, 102 invited lead key note lectures, and 279 abstracts in seminars, symposia, conferences. Uh, he has to his credit. And Dr. Srivastava has been awarded SN Ranade Award for Excellence in Micronutrient Research, FAI Silver Jubilee Award, International Plant Nutrition Institute FAI Award, Netaji Subhash Chandra Bose Award for Excellence, National Magnum Foundation Award for Excellence, Dhirubhai Morarji Award, BL Jain Award, World Aqua Foundation Award, Best Citrus Scientist Award, in addition of his biography, which has appeared in two world famous biographical compilations. Dr. Srivastava is a life member of 33 academic societies, besides honorary fellow of World Association of Soil and Water Conservationists, Society for Horticulture Research and Development, and Society for Biotic Environment Research. He is author of books like Citrus, Soil and Climate, Citrus Nutrition, published by IBDC Lucknow. And he is also editor of books entitled like Advances in Citrus Nutrition by Springer Verlag, Netherlands, and Fruit Crops Diagnosis and Management of Nutrient Constraints in by Elsevier, USA. He is inducted as Fellow of 11 professional academic societies and Honorary Fellow of 3 academic societies, member of Asian Council of Science Editors at Seoul in Korea. Dr. Srivastava is also editor-in-chief of research journals on earth sciences and International Journal of Horticulture and Crop Science Research and Current Horticulture. Besides, he is also honorary editor, regional editor, executive editor, member of associate editor, and member of editorial board of 4 international journals and associate editor of also of 4 international journals and 20 journals of national repute. Dr. Srivastava is a regular paper setter with 9 agriculture universities, and he is member of the management committee of 4 ICR based organizations. Besides, he is also adjunct faculty of 3 agriculture universities and visiting professor at 2 universities in China and a visiting scientist in Iran. Dr. Srivastava has held foreign assignments as a citrus expert to government of Nepal, and he has been keynote speaker or lead speaker in various conferences, congresses in China, in Iran, and Israel. So, with a lot of experience, viewers that you have heard, and the wide, talented person that we have, Dr. Srivastava, with wide and long experience working on citrus soils, especially, would be linking us from the soil health with the human health that is very important in today's world through fruits. So, not taking much time between the viewers and the speaker, I would like to invite Dr. Srivastava to deliver his talk.
Thank you. Thank you, Dr. Kalia, and thank you, Professor Brahma Singh, sir. Very good evening, friends. Indeed, a pleasure talking to such a august gathering online. As I will be talking today on a very interesting topic, and hopefully you will enjoy listening our content. The topic is the contribution of soil health to human health through food crops. Well, friends, if you look through the historical perspective, the fruit plants are our greatest heritage, and they have had a relationship with the prehistory. How, where, and when the domestication of these food crops occurred is being slowly revealed these days. However, the domestication of food crops, they have taken place at a much slower rate compared to the domestication of pulses and beets. Of course, the domestication of food crops came much later, only after the domestication of cereal crops and pulses. Development of food culture in the Fertile Crescent evolved at 2 major loci, the Tigris-Euphrates civilization of Mesopotamia and Nile Valley culture of Egypt. These are the 2 major centers where the real culture of the fruit started, of course, in a wide format. Needless to say that the fruit crops are pretty rich source of all kinds of nutrients. I mean, you name a nutrient, you name a vitamin, you'll find them in the fruits depending upon the type of the fruit, temperate, tropical, or subtropical. If we go through the historical perspective of the soil fertility, the cultivation of plants for the food, in fact, allowed that humans to convert from the nomadic hunting to gathering of the lifestyle to a settled experience. Now, if you further trace back that how the soil fertility and the human settlement, they co-evolved together. In fact, through the history, you will find 3 major basins: Amazon basins, Nile basins, and Indus River basins. These are the 3 major river basins which are known as major civilizations which settled along these rivers only, for the simple reason that the soil along these rivers were quite fertile, and human settlement always preferred where there is water, where there is a fertile soil. So that's no exception. Now, how the soil fertility concept, in fact, started, it dates back to some 2500 BC, where the fertility of the land was mentioned, and the fertility of the land at that point of time was mentioned that how the periodical flooding that was able to enrich the soil with the annual flooding of those nutrients. And then, of course, later on, the theory on the green manuring started, then humus theory started. In fact, the basic flaw of this humus theory was that at that point of time, it was believed that the carbon for the plants, they are coming from the humus. Of course, later on it was refuted. And then the soil fertility and plant nutrition concept, it came later on, where the water was found that it is the sole source of nutrients. And unless until there is water, nutrients will not be able to reach up to the plant. And then, of course, the book on the horse hoeing husbandry, and they developed the concept of hoeing and seed drilling concept. And then later on, the modern concept of soil fertility and plant nutrition started ever since the various concepts by Liebig somewhere in 1800 to 1900, they were floated. And those concepts of Liebig, in fact, it floated a concept of a yield pilferage theory. Yield pilferage theory means that whenever there is some imbalance in the plant nutrition, there is always likelihood that the optimum productivity of the food crops or any crop will not be achieved. So if at all there are, say for example, 3-4 nutrients which are deficient simultaneously, the nutrient which is most deficient, that will dictate the very productivity level of the plant. So that was the concept of Liebig at that point of time, and this concept stands valid even today. And all these concepts, in fact, laid the foundation that whenever there is a healthy soil, mankind is always at ease. One can think of very nicely. If there is a good nourishment of the human being, probably the brain acts beautifully, and that's how the human population evolved. And that's how the good soil fertility, the good plant health, and the good human health, they all three co-evolved together. And while exploring the 3H webnet, there is the interconnection of the soil health, plant health, and human health. And then, of course, when there is a good synthesis of all these three concepts, there's always a different kind of happiness to mankind. And of late, this was the reason that how the stripping of nutrients started taking place at the global level. There's a huge inflow of human population from rural areas to the urban areas, and that's how the nutrient stripping also started. There's a huge consumption of disparity in the soil nutrient deficit distribution versus human nutrient consumption, and that's how the nutrient deficit started, and that's how the nutrient stripping started. And we started saying that probably that's the starting point of nutrient mining, and that went very unnoticed to the human population and to the orchardists of the practitioner. As long as they cut in the productivity level, we started facing, and with fruits no exception. Now, there's hardly any evidence to prove it, but of course of late there were some exercises that unless until you evaluate them on a spatial scale, on a temporal scale, and as well as on a spatial scale. So this is the one exercise which I wanted to prove that these are some 7 crops, right from apple, citrus, strawberry, tomato, lettuce, carrot, and peach. And there were some sites which were selected, and 1963 it was the base value was taken, and then on the same site in 99 it was taken. So these are the 3 nutrients they tested: calcium, magnesium, and potassium. And to your surprise, within 35 to 36 years of time, there was a huge reduction. In tomato, there was a reduction in the calcium percentage by 61%. And likewise, there was a reduction in the magnesium in the apple, carrot, peach. So over a period of time, these nutrient concentration in a spatial mode as well as in a temporal mode, they started declining. And what was the reason? The reason was that we were not in a position to match the amount of nutrient that is required by these crops versus the amount of nutrient that we had. Probably at that point of time, there was no modus operandi available to really gauge that what amount of nutrients they are being transported. Because we hardly believe in a system that if at all you have a certain productivity level, we hardly bother that this much amount of nutrient which is transported out of your production system, and unless until that much amount of nutrient which are drawn by those crops, they are returned back to the soil, probably will not be in a position to strike a balance between the amount of nutrient which is drawn versus the amount of nutrient which is added over a period of time. And let it be any print media, let it be any electronic media, let it be any social media. We just keep talking about the galloping nutrient mining in these food crops. And these nutrient mining causing multiple nutrient deficiencies, somewhere cutting down the crop carrying capacity of the soil, which we don't realize it unless and until it becomes too late and too little. So this is how we started facing them. And then that led to the concept of nutrient density standards. Instead of talking about the nutrient concentration, this was the basis that why these days we keep talking of the nutrient density. And nutrient density is nothing but the nutrient concentration vis-a-vis the energy level. So one, how much calorie that you take, and with this calorie to fruits, how much grams, how much milligram of the nutrient they are delivered into your system, and that really gave us a concept of nutrient density standards. And this was the reason that why even today at the global level there is a dietary deficiency to the extent of more than 60% dietary deficiencies in terms of iron, more than 30% in terms of zinc and iodine, more than 50% population is showing the dietary deficiencies in terms of sulfur, apart from the calcium, magnesium, and copper. So there is hardly any nutrient which is not being translated into the dietary deficiency. It means whatever foods that we are consuming today, they are not able to meet our dietary requirement. Dietary requirement, of course, the energy requirement is able to be fulfilled. But at the same time, since because of the lower nutrient density, they are not able to meet out your nutrient requirement. So that's how the kind of nutrient deficiencies we started facing. Now it's needless to say, when you take a simile between the plant and the mammals when I'm talking of human beings, there's hardly any nutrient which is available in the plant and they are not required by the mammals. So there's a great amount of consonance between the requirement of different nutrients by the plants versus the requirement of different nutrients by the human being. It's always said that there's a healthy soil, there has to be a healthy plant, and if there is a healthy soil, there's a healthy plant, and the human being has to be healthy. And that's how the concept of microbes, they started, as our Professor Singh was talking in the introductory lecture, that there's a kind of similarity in the microenvironment between the glut, between the rhizosphere, and between the plant. And that's how the man, soil, and the plant, they co-evolve together. I'm just trying to give you some empirical evidence that just imagine the stomach, there's acidic soil in the acidic environment in the host environment, and in the soil there is the acidic soil here, and the macrobiota. Duodenum, neutral soil, neutral pH, rhizosphere, there is a neutral pH, and that's how there is a similarity between the microbial diversity. Colon, neutral pH, endosphere, neutral pH, and that's how these endophytes, once you consume these foods, the endophytes which are embedded in these foods, they go inside the body, and that's how those beneficial endophytes they constitute a part of microbiota, and that's how these macrobiota, they are often, it is said that they are responsible for your health. And if at all there is an attack by the pathogen, either in a human being or in a plant, there is a distortion in the microbial fabric of that rhizosphere soil, or even the glut microbiome. That's how your resistance is compromised, and that's how you have a reorientation in different microbial activity. In fact, if you look at the diversity in the soil, it's the biggest gift to the mankind. Just imagine, I just keep saying that the potato is grown in alluvial soils, and potato is grown in the black soil. Alluvial soils are coarse textured, black soils are finely textured. Look at the amount of tuberization. The amount of tuberization in alluvial soil is much more. The quantum of potato that you harvest in the alluvial soil is much more compared to the quantum of potato that you harvest are the rate of tuberization that you notice in black soils. What are those reasons? Why there is a good flavor of those potatoes? Why there is a greater concentration of potassium in the potato which is grown in alluvial soils compared to black soils? Look at the classical example of alphy soils. Alphy soils, they are basically acidic soil. Look at the small cardamom which is grown on alphy soils of Sikkim. They are almost similar to the alphy soil which is grown in Kerala, the large cardamom. What are those defining factors? Even after putting up 32 years of service in a crop like citrus, if you ask me that what are those micro level parameters which are responsible for development of such a exquisite the flavor of Nagpur mandarin, sometimes you feel like just peeling off a Nagpur orange, even in a train, and something like it moves, it gives a fragrance to the entire compartment. So that's the beauty of the soil. But still, we are not in a position to decode the very nature and the property, the very constituent of soils which are really responsible. Of course, it is in combination with the climate that are able to give you a very exquisite quality development, how the flavor development, and that's how I always keep saying that fruit crops, whenever we talk of the fruit crops, we hardly evaluate the nutritional quality of those fruit crops in terms of the qualities which really define the consumer acceptance. Usually, we analyze the quality of the fruits in terms of the TSS, acidity, juice content, which we hardly do it at the consumer level. Consumer has to see that these are the fruit quality parameters, and whenever we are evaluating any input response, these qualities have to be considered. Now, apart from that, soil is also, unfortunately, of late, it is being abused because of the heavy use of agrochemicals, and these pesticides are one factor where the soil health is compromised. When soil health is compromised, keep in mind that there is every likelihood that the plant health is also compromised. WHO has identified these are 23 major soil contaminants which are affecting not only the soil health, but at the same time somewhere the plant health, and if soil health and the plant health both are compromised, there is every likelihood that the human health would also be compromised. There are certain negative effects of the soil on the human health in terms of the heavy metals, in terms of the organic chemicals, airborne dust, soil pathogens. For example, the soil fungus Ixarohelium rostratum was responsible for fungal meningitis outbreak in USA, and that came from the soil only. But these are some of the examples of negative effect of the soil. But just look at the antibiotic industry globally. As much as 40% of antibiotics, they are derived from the streptomyces, and some 60% of all anti-cancer drugs, they come from endophyte fungi belonging to the genus Texas. Now, such is the magnanimity of the soil and the microbes. Look at the constituent of soil, the clays, kaolin, which is a non-expanding clay. They are responsible for anti-diarrhea and anti-ulcer drugs development. And something like smectite, which are known for the detoxifying the aflatoxin. Aflatoxin is nothing but the mycotoxins which are produced by the various kinds of molds. So somewhere these soils have really revolutionized the human health, directly or indirectly. And if you trace the history of the soil health, of course, of late we started some 25, 30 years back, only we started talking of soil health. Otherwise, the history of soil health is somewhere 100 years old. And somewhere in 1910, the soil health term was used. And the soil organism was for the first time was used in 1931. And that's how they highlighted, Stanford highlighted for the first time that soil health and national wealth, they are complementary to each other. We also tried to see that on a Google search between 1850 to 2000, that is there any issue of soil health? Now, to your surprise, the soil and the health, they are defined as two independent terms rather than soil health together. And the soil health together, it came into existence only recently, ever since we started abusing our soil health with the indiscriminate use of chemical fertilizers, with the indiscriminate use of various kinds of agrochemicals. And then, of course, soil health and the soil quality, they are often overlappingly used. And the soil health, then later on, of course, this is a big event these days, soil health assessment. What are those concepts? What is the concept of soil health? How these are assessed? How these are managed? And when you talk of soil health concept, you have to define the soil health. And when you have to define, you have to interpret the soil health. When you are talking of the assessment of soil health, what are those indicators of the soil health? These are the indicators of the soil health. And then what are those methods through which you can really evaluate the soil health? Soil health card is one these days. The government of India has already taken a massive initiative in the last somewhere 6 years of time. Nearly 1.3 or 1.4 million soil health cards have been released. And these are the principles of soil. When you talk of the management of the soil health card, these are the principles of soil management, and these are the practices of soil health card. So it's a complete concept of soil health card. And soil health card, because of the very precise initiative of soil health card by the government of India, of late we have started realizing the impact of soil health card. There is already cut down in the fertilizer use, of course, regardless of the fruit crops or regardless of crops. This is already cut down in the fertilizer use by some 8 to 10%. There's an increase in the crop productivity by 10 to 12%. But that was the first phase of the soil health card. Soil health card these days now in the second phase. Now it comes to the precision. That time has come that let us talk of the crop-based soil health card. We are pretty good in soil analysis. We are pretty good in leaf analysis, but we are equally poor in the interpretation of soil test rating. Even now, to your surprise, friends, the soil fertility constant, they are diagnosed using the soil fertility rating, what is given for paddy and the wheat. Just imagine, there's hardly any systematic effort that we have yet made the crop-based soil health card, and foods are no exception. If at all the horticulture has to strive on a sound scientific basis, we will have to work very seriously on crop-based soil health card. A soil health card which is made exclusively for citrus cannot be good for the mango. A soil health card exclusively made for mango cannot hold good for lychee, and that's how our soil fertility constraints are wrongly diagnosed. So what happens that instead of nitrogen you add phosphorus, instead of potassium you add zinc, instead of zinc you add iron, and that's how the results of the soil test rating, it keeps on giving erroneous results, and we hardly get the response of the crop. And more importantly, of late, what happened that these soil test rating, they have never considered that the yield level. And any soil test rating that has to come yield level. This is where we have taken a massive exercise over the last 32 years, taking citrus and gauging the citrus almost every nook and corner of the country, be it a Nagpur mandarin in central India, be it a Kino mandarin in northwest India, be it a Khasi mandarin in all 8 northeastern states, Mosambik sweet orange in the Marathwada region of Maharashtra, the Sadgudi sweet orange in the entire southern India, the Malta sweet orange in the entire northwest India, and the Uttarakhand, and the citrus aurantifolia, almost the entire south, along with the western Maharashtra. We have generated a huge database. We have analyzed in the last 32 years nearly 30 to 35,000 soil and leaf samples. And based on these massive database, we have come out with a Mutant diagnostics based on the soil fertility and leaf analysis program. And over the last 20 years or so, after staying at Nagpur, we have been launching the leaf analysis program and the soil testing program and the soil health card exclusively for the citrus on a countrywide basis. And then we also have a modus operandi by application of spatial technology in the north. For example, the northeastern India, we identified as many 52 sites across 8 northeastern states covering some 102 orchards, and we identified that there could be some of the orchards where there is no zinc deficiency, magnesium deficiency, phosphorus and nitrogen deficiency. And these are the areas where there is no nutrient deficiency. So this also gives us a kind of nutrient modeling, that using these spatial tools and using these nutrient diagnostics, you will be in a position to identify that what are those areas where there is no nutrient deficiency, or what are those areas where there is a nutrient deficiency. So citrus or any other food crop being a perennial in nature, you can really practice those fertilizer recommendations so that you are able to address those fertilizer constraints right at the time of planting, rather than growing a citrus crop or any other food crop and later on experiencing those nutrient deficiencies, experiencing the cut down in the productivity, until then probably it will be too late, because these all these perennial food crops, their juvenility is too long and they take at least 5 to 6 years before they flower. So that sort of exercise and the concept we have to bring into the picture. This is the an example of soil health card, how we developed the soil health card for citrus, staying at Nagpur. And this kind of soil health card, I request our all researchers, whosoever is listening to my presentation, that let us work seriously, let us work unitedly for the soil health card for food crops or for any horticulture crop. Unfortunately, this is the most missing link. We do not have a crop-based soil health card for any horticulture crops, with an exception of citrus, to some extent mango, could be some other crops also, but I hardly see that there is any comprehensively defined crop-based or the food crop-based soil health card. And that kind of exercise is very much required if at all you are trying to infuse the balanced nutrition, to infuse the nutrient density, to infuse the longevity of these orchards, to infuse there's a good amount of nutrient along with the calorie, so that whenever it is being consumed by the human, there's every likelihood that you have good health of the human being. Now, friends, this is another very massive exercise that we do. You have a soil test rating, and there is some productivity level. For example, here I have shown that here's a target level of 10 tons per hectare. You have a soil test rating, for example, the nitrogen 100 kg per hectare. I want to raise my productivity level from this 10 tons to 20 tons. How much nutrient is to be added? And this kind of tripartite relation for any food crop has been developed for the first time. I take pride in communicating with you that this kind of ready reckoner for the decision support for the fertilizer recommendation for Khasi Mani we have developed for Nagpur Mani also. This is the one example I am trying to collect. These are very much required, that you have identified the fertility constant, you have identified the nutrient constant. What are those modus operandi through which you can really ensure the balanced fertilization, and then you can ensure the arrestation in the nutrient mining, and then you are able to prolong the productive life of these orchards. So that's sort of premise we need. One of the major constraints we have been talking of late, whether it is food crops or vegetable crops or any horticulture crops, although the rhizosphere of horticulture crops, they are pretty good in organic carbon content, but you keep on applying farmyard manure or any organic manures in terms of the vermicompost, poultry manure, neem cake, green manure versus inorganic fertilizer. There's hardly any change over the last 2004, 8 years of time that we evaluated continuously, we kept on applying. For the simple reason that this kind of addition of organic carbon is more into the active fraction of the soil, but compared to the passive fraction of the soil carbon stock. And unless until whatever carbon that we add to different manures, whether organic manure or green manure or any other practices, the carbon that is experienced into the soil, that has to be lost into the passive pools, because that passive pool carbon is more recalcitrant in nature, and more recalcitrance means that carbon is more resistant to further decomposition. So unless until these practices and they lodge carbon into the passive pool of the soil, probably will not be able to derive the potential benefit of this organic carbon, or something like organic culture of these crops, and fruits are known for organic culture, but this is where we have been utterly failing. What are those options that we have in order to put carbon into the passive pool, put carbon into the legacy carbon, so that once you put that carbon into the soil and grow fruit crops, and for generations that carbon is available for the betterment of the fruit crop. And you know it pretty well that once you are increasing the organic carbon content of the soil, there's every likelihood that you will be increasing the nutrient pool of the soil. You will be creating a nutrient sink into the plant, nutrient sink into the soil. If there is a nutrient sink in the soil, there is a greater nutrient sink in the plant also you'll be creating, because you know it pretty well that carbon nitrogen ratio, carbon phosphorus ratio, carbon iron ratio. So whenever there is an increase in the carbon pool of the soil or the carbon pool of the plant, there is likelihood that you will be creating a parallel nutrient pool into the plant also. So indirectly, any practices that is able to increase the organic carbon and the carbon content of the soil, every likelihood that will create a parallel nutrient sink, and thereby there's every likelihood that the nutrition quality of those crops will be good enough. One of the prudent options that we see in order to increase the organic carbon content is the mycorrhizae. Unfortunately, in a country like ours, they are very limited experts on mycorrhizal research. But mycorrhizas are known to increase the nutrient absorptive power of these root hairs to the tune of 20,000 to 50,000 times. It is a historical record that these once there is an inoculation of these mycorrhizae, they are able to increase the surface area of these root hairs by 20,000 to 50,000 times. And just imagine it comes because of the glomalin, which is a glycoprotein, and it offers somewhere it has got some 30 to 40% of glycoprotein, and it offers some 10 to 15% of recalcitrance form of the carbon. And this is where a very famous liquid carbon hypothesis was launched by Professor Christine. And liquid carbon hypothesis is nothing but once you put a mycorrhizae into these fruit crops, the whatever carbon which is being accumulated to the external hyphae of these mycorrhizae, at least 10 to 15% of that carbon is translated into the roots of those fruit crops, and thereby those fruit crops become more resilient. They become more healthier. They become more nutrient rich. Another option that we have is the biochar. Biochar, you know it, that the terra preta concept of Harappa concept, that mummified plant, they were able to produce those biochar. And biochar, can you believe it, that once you add biochar, which is basically plant origin, of course, any kind of plant origin you can convert it into biochar, depending upon the temperature of the pyrolysis, the feedstock material, nature and properties of the feedstock, whether the oxygenation is to what extent we are using it. Depending upon that, the quality of biochar you can produce. And biochar recovery, the carbon recovery is somewhere 40 to 50%. But the problem with the biochar is that, the only problem is that biochar has got a very high rate of recommendation, first thing, and secondly, it takes some time to really put that kind of physical, chemical, and biological effect on the biochar. But the biggest plus point of this biochar is the carbon residence time. The carbon residence time of biochar is somewhere 100 to 150 years. You just imagine, once you add biochar into the soil, into the rhizosphere of any fruit crop, it is a one-time treatment. One-time treatment. Your soil will be healthier with respect to carbon enrichment for nearly 100 years. It's such a magnanimous recalcitrant form of carbon it supplies. And these days the small scale biochar machines have started coming up, pyrolysis concept is coming up. But the problem is that, looking at the very small land holdings of biochar is still a big question mark with respect to very fast release of carbon dioxide into the atmosphere. Some of the toxins, they are also expected that they are released into the environment. So that environmental issue is yet to be solved. Otherwise, biochar happens to be environmentally very benign concept. Most of the studies, they establish this fact. The next question comes that is there any macro which can really unlock the productivity potential of these food crops? Probably this was one of my own reviews. There's hardly any food crop where these macros, I mean, I'm talking of the rhizosphere macros, I'm not talking of the endophytes macros. So these rhizosphere macros could really unlock the productivity potential of these crops. And not only they unlock the productivity potential of these food crops, regardless of the nature and properties of soils and regardless of the climate, but they also prolong their productive orchard life. And there is a direct evidence that these are the macros which enter into the body of these plants, and then this is how these endophytes, these macros also act as endophytes, and when you consume these fruits, these endophytes go into your body and become a part of your gut macro, all beneficial ones, and thereby it offers a resistance against any of these pathogens, against the diseases, and that's how it offers the health to the human body. Well, friend, this is another very massive exercise that we finished this while staying at Nagpur. The time has come. Gone are those days when you used to talk about the application of 1 macro, 2 macro, 3 macro, something like that. A time has come that probably instead of applying those chemical fertilizers like nitrogen, phosphorus, potassium, zinc, iron, probably the time has come that when there is a requirement of nitrogen, you add a nitrogen fixing macro. When there is a requirement of phosphorus, you add phosphorus solubilizer. When there is a requirement of iron, add iron solubilizers. When there is a requirement of zinc, you add zinc solubilization. So it means that you can very well partition different nutrients. You can schedule nutrient requirement according to these macros, and these macros could well supplement these nutrient crops, apart from the resilience against the carbon dioxide released from the atmosphere, whatever carbon that you are adding into the soil. So these are, and then the another biofertigation. These macros can very well be rooted to the fertigation, and let us create a kind of blanket of those macros into the soil, and do not allow those chemical fertilizers to react straight away with the soil, and let these blanket of macros come into the picture, and they interact with the macro, and thereby they act as a nutrient sink, and thereby they neutralize the very exothermic reaction of these nutrients before they really react with the soil, and that's how they ensure the greater nutrient efficiency. So these are the enormous possibilities of macros in food crops. So what we did while staying at Nagpur Citrus Center Citrus Research Institute, we developed a Microbial Consortium. And consortium of these five macros. One is the asymbiotic nitrogen fixer, and there is silicate solubilizer, and there is phosphate solubilizer. All three are phosphate solubilizer. And apart from these, there is every likelihood that some of these macros, they are very strong antagonists against soil-borne pathogens. And this is how a liquid formulation we have developed. And whatever disease planting material that we are producing, some 2 to 5 ml of these microbial consortium we are adding into each of these plants. And whenever you put them into the new area, there is a massive cut down in the mortality of these plants. And also the native macros along with these plants, there is a native macros that are being built up, and the plants are at ease in the new microbial environment, and thereby there is a much reduced mortality of these food crops. Citrus is one, of course. And then we have gone a stage further later. We have developed a microbial consortium, these pellets. These pellets are nothing but something like acrosin tablets. And probably, who knows, in the years to come, probably maybe in another 2 or 3 years, we will be in a position to give these microbial consortium tablets, pellets to the farmers. So instead of, you add some maybe 500 to 1 kg of these biofertilizers, let us add 1 or 2 pellets and just put it into the rhizosphere soil, and the job is done. And moreover, the pellets of these macros, they release these macros very slowly, rather than these macros are exposed to the extremes of the soil conditions, whereby they are inactivated. So this is another advantage, and probably we will also be trying to do some kind of capsuling also. In years to come, we could have microbial capsules also. So these are the enormous possibilities of macros. And these macros, we also tried that to what an extent whether these macros are really effective against the carbon sequestration in soil. To your surprise, there was much less carbon dioxide release wherever we have added this Microbial Consortium, what we call integrated soil fertility management system versus recommended doses of exclusive chemical fertilizer. There was some 30 to 40% reduction in not only in the fertilizer requirement, 30 to 40% reduction in the fertilizer requirement, there was some 30 to 40% reduction in the carbon dioxide release. What does it mean? It means that these macros, whatever we have identified in a complementary mode, these macros are very effective against the release of carbon dioxide. It means we are able to lodge that carbon into the passive pool. Otherwise, had it been an active carbon, that would have been lost through the carbon dioxide. What we are noticing here in terms of the chemical fertilizers, these chemical fertilizers are, although increasing the yield, there's a yield increase, means there's an increase in the root activity, there's a root yield also, there's a root flush, then only there will be flush in the leaf. So the carbon which is added to these chemical fertilizers, they are mostly act in the active pool, whereas the carbon which is added to these macros, they are mostly in the passive pool. And that's one of the reasons why we have a reduced carbon dioxide release from the Microbial Consortium treated integrated soil fertility management. Well, friends, we often keep talking. Say, for example, we take very much pride that our entire horticulture production has surpassed the food and production somewhere by 28, 30 million tons. Fine. And they all credit goes to, we are all researchers which are within the ambit of horticulture crops. But the time has come that let us talk in terms of the carbon footprints. When there's growing consciousness about the climate change neutrality, let us develop some crops which are climate neutral in nature. Let us develop those package of practices which is climate neutral in nature. Let us develop those fertilizer practices. Let us develop those irrigation practices. Let us develop those all soil plant health management practices which are carbon neutral in nature. Is there any possibility that while increasing the carbon footprint of these crops, you can really do carbon trading? There is a possibility. There is a strong possibility. This is one of the examples that compared to the woodland, the growing mango was giving a much higher cost of the carbon, which was sequestered into the soil. And in 100 years of time, there was of course, it was done in a modeling. The BC ratio was somewhere 1:4. So it gives us a clue that why not talk that this is the carbon footprint of our food crops, and why we say like this. A fruit crop which grows for 40 years, 50 years, don't you feel that we are creating a parallel perennial carbon sink in the perennial framework of these food crops? 50 years, 60 years, it is surviving. Apple surviving for such a long number of years. Mango, 60 years, 70 years is so common and giving you a perennial framework. If you are, if you are sequestering carbon through a forest land, fine. There is no ecosystem service provider. There is no service provider to the mankind. It's environmental factor. But when you grow a crop like mango, citrus, I mean, just imagine you are storing the carbon into the perennial framework of these crops. At the same time, you are getting the additional yield, and with this, you are creating a nutrient sink. Plus, you are creating a carbon offset technology. So this is what is called carbon neutral technology. Why not to compute a carbon footprint of our entire food industry? Go to the international market and say, this much is the carbon trading, this much is the carbon neutral mechanism that we have developed through these food crops, and say that this is how we have cut down the carbon load of the atmosphere. This is how we have carbon offset technology to these food crops with the use of certain benign package of practices. Unfortunately, this is where we are missing our trait. This is where there's a time that we have to vouch into these dimensions of climate change, using food crops, using human health as one of the prime triggering factors. And the concept that these days we are very massively advocating is the rhizosphere hybridization. You must have seen the fruit crops, see the ficus trees, all people, bargad, you see the neem trees. Have you seen any disease in the neem plant? Have you seen any disease in the ficus plants? You hardly see any disease. So it means there is a resistance, there is a natural resistance, and from where this natural resistance is coming up, this natural resistance is coming to the rhizosphere macros. So what we did, we just took the rhizosphere of that ficus trees and put it into the rhizosphere of citrus. And to your surprise, all what we call the rhizosphere hybridization. We came out with the concept. This is a part of one of my PhD student, the rhizosphere hybridization. So we took the rhizosphere of the soil and put it into the rhizosphere soil of the citrus, and there was an elevation in the enzymatic activities of this hybridized soil. And this hybridized soil was not only with respect to the increase in the enzymes was observed, there was a much better nutrient pool, there was a much better nutrient supply chain, there was much higher amount of organic carbon, all together contributing into much higher yield. And just imagine, hardly 1% of macros we are able to characterize, but 99% of those macros which are yet we are not in a position to characterize by any means. 99% of the rhizosphere macros which are culture independent. Now with these culture independent macros we have added into the citrus rhizosphere, and such a beautiful concept of rhizosphere hybridization has come, and thereby this hybridization could add another level of resilience, another level of plant health management. In the time to come, probably this is going to be the core frontier area of soil health management. And thereby another frontier area, that plant nutriomics. Just imagine, we add a nutrient which is acting at a cellular and subcellular level. You just imagine how a plant senses the nutrient deficiency. Every root hair, root cell having plasma lemma is having a nutrient density, and that nutrient sensors, they are made for each and every nutrient. When there is a nitrogen deficiency, a plant will trigger of its nutrient sensor for nitrogen, which is nothing but a protein, and that will sense the availability of nitrate and ammonium, and it will make some certain kind of root secretion, rhizodeposition, and that's how the nutrients are attracted. In the introduction of a concept like genomics, transcriptomics, proteomics, and metabolomics, probably will be adding another dimension, that how once a plant absorbs a nutrient, how these plants are mobilized, uptaken, translocated, and utilized at the site of utilization, where the fruits are the premier sites of those. Now, this is adding another dimension of nutrient use efficiency, and in yesteryears we have been able to fail very utterly. Now, this is added another dimension, that that a nutrient which is acting at a cellular and subcellular level, we are evaluating them at the whole plant level. Just imagine a nutrient which is working at a cellular subcellular level, we evaluate them. What is the yield per tree basis? What is the yield per hectare basis? So how much dilution that we are making? This is the biggest drawback of our agronomy-based evaluation, that any stimulus that is acting at the cellular and subcellular level, unless until we evaluate them at the tissue level, at the plant part level, probably will not be able to make much headway about the underlying mechanism involved with the use efficiency of those nutrients. So probably in the years to come, plant nutriomics and another very budding phase of plant nutrition is coming up. Well, friends, your story of adding soil health will not be complete if you have added some plant growth promoting macros into the soil. At the same time, there are soil-borne pathogens. What will happen? Those soil-borne pathogens will neutralize those plant growth promoting macros. So the soil health and plant health care, they have to go together, and they have to go together through a consortium-based macros. This is how one example we have shown here in a citrus. The first stage what we are doing, the isolation and characterization of soil plant macro, and then we are bioprospecting the soil plant health consortium related consortium mode. So we are developing a consortium of plant health, plant growth promoting macros, consortium of microbial antagonists, so that the plant health, so that the plant growth and the soil health, they join hand together, and soil health, plant health, they together act. And then at the same time develop a consortium of endophytes. Some of the diseases, like I have taken a citrus bleeding disease, which is a very lethal disease these days globally, so that you are able to develop that consortium of endophytes, and those endophytes neutralize the that pathogen inside the plant. And then you have certain nano composites of nano zinc and iron, whatever nutrient content that you have, so that whenever these pathogens, they block the nutrient supply chain, you are able to create a parallel nutrient supply chain. So combination of consortium of these macros plus the nano composite as a parallel nutrient supply chain, they will create a very well redefined, revisited production system. Not only is it such examples can very well be replicated in other food crops, so that soil health, plant health go together in hand to hand. Now, it's needless to say, it's a web network of various kinds of soil constituents versus the plant constituents, all together working in a tandem, and then working for the plant health. But still, we know very little about the constituents between the soil, how they are interacting with this pathways by which soil constituents move into the human body, the mechanism by which different soil constituents, once enter into the body, can harm or benefit the human body. Still we do not know. Still we are in a very rudimentary understanding about that. And therefore, there is multidisciplinary research is warranted to facilitate the discovery of new antibiotics and a greater understanding that how these soil materials added to the soil, they are used for the food production, affecting the health, and thereby deciphering the complex relationship between the soil and the human health. This is what the meaning comes out. These are the only four futuristic views. Of course, it is futuristic, socialistic nature is more increasing the knowledge of the soil condition. Still, our knowledge with respect to the nutrient supply chain to the plant vis-a-vis human. So, soil health, plant health, and human as a continuum is still a big question mark, and still we have been little under stress. We have yet not been able to, although there is a consensus, there is understanding, but still we are not able to really decode that coordination between the three niches of nutrient supply chain. Then the emerging contaminants. We have to develop certain kinds of new tools and remediation methods also, that how the soil health of late they are coming at a stage with respect to various kinds of agriculture practices. We need that part also to be robustly developed. Increasing the connectivity and codification related to the soil contamination, we do need on a very urgent basis. And then, of course, increasing the knowledge of the soil capital. As I've already talked about, the increasing the knowledge about the carbon capital, nutrient capital, carbon footprint. And let us go another dimension, water footprint. This is another dimension, because water and the nutrient, they go hand in hand. So these are some novel options that we have. Somebody said our very famous industry that eat your food as your medicine. Otherwise, you have to eat medicine as your food. With this message, I thank you to all of you, and hope that you must have got some points and some research agenda for the future. Of course, I have thoroughly enjoyed. Let me express my thanks to Padma Singh, Professor Brahma Singh, Dr. Pritam, Kalia, Dr. Shailendra, Rajan, all three for giving me this opportunity and sharing a concept which is absolutely fantastic to talk. And at the same time, we have also shared what we have been doing over the last 32 years while staying at Central Citrus Institute, Nagpur. Thank you very much.
Thank you, Dr. Srivastava, sharing your very rich experience and the experience from the literature what has been reported on the topic. Dr. Kalia, could you just highlight few points of his talk. Few.
Yes, sir. Thank you, sir. Sir, I think we have had a wonderful talk in a different mode. Uh, that it is the soil which is actually where our plant stands, the root stands, and everything comes through the soil. Normally, we talk about the plant on the top which we eat, but where it comes from, it's today that Dr. Srivastava has explained in detail about all aspects of soil and how it's going to be in futuristic way. That always we talk about from the plant improvement point of view, that futuristic plant development, how it will go, how we'll be able to go forward. But he has connected everything with the soil, that how in future we are going to have a relationship with the new plant improvement technology linked with the soil, because ultimately all nutrients has to be rooted through soil to the plant. So soil importance has been highlighted in detail, right from its historical perspectives, giving various examples of history, how the soil importance has been realized uh, over the years, and in the recent past, the emphasis has been more directed towards the soil. And also, he has mentioned that the decline in the nutritional value of our fruit crops and in other crops also that we find, and that is the major region that we find that especially in Southeast Asia, our population is suffering from deficiency, malnutrition, undernutrition. It's actually linked with the diet, and the diet is plant which comes, the nutrient comes through the soil. So it's a very important for us to realize those things. As in the past, not much nutrient used to be put in the soil, but the management of soil has been realized in the recent past, and also has been highlighted that the government of India has gone because of that reason for soil health card. And on the carbon footprints, because every time it's talked about that, what is the carbon level of the soils, what is the productivity we are talking about, it's low, we say it's low, how to improve it. So all those aspects, carbon footprints, and also he has talked about the water and the other various aspects which are not much looked into, and endophytes, which is very important aspects, and the soil diversity that he has made mention of from that point of view, and also mentioned that there are some problems, that soil contamination, which also pose threat to us. So how to get rid of all those aspects that he has been highlighting in detail. And not about the soil health card, but he has highlighted that we should have, for increasing precision, the crop based soil health card, which is also being looked into. And he has very rightly given examples of what work they have done at Nagpur, Mutant Optima based diagnostic for different citrus cultivars, that he has mentioned in detail and highlighted the things. And also he talked about mapping of different sites from the nutrient point of view, which will be good for nutrient modeling, Kasi Mandarin, especially mapping of soils he has mentioned. And the constraints with respect to the carbon, he has mentioned that calcitrant and recalcitrant aspects, and in that, which is important aspect, I think, which has been in detail he has talked about. And I think the viewers must have been educated. If some students who are studying soil science and holistically any crop science, they must have been educated with this aspect, that mycorrhizal sphere glomalin he has talked about, which is very important aspect. Actually, we have also been highlighted along with the viewers. We have been educated through these aspects, how to the carbon sequestering, and with respect to that, how to reduce the carbon release into the atmosphere, and sequester it, and also the use of biochar. He has in detail mentioned about. and then unlocking productivity through plant microbe synergy, Microbial Consortium, and rhizosphere engineering, which is very important aspects that the soil is talking about the engineering aspects also, and also he has gone towards that what are the futuristic aspects where nutrient use efficiency can be enhanced with the microbial consortium that they have developed from the nitrogen fixer point of view, silicate solubilizer, phosphorus solubilizer, etc. And they are anticipating. He has also mentioned about that these microbial tablets and release of microbes slowly, microbial capsules especially, which are the futuristic aspects to go about with these all important things. And the field evaluation of microbial consortium, where he has laid emphasis on the reduction of carbon dioxide released into the atmosphere, and uh, reducing the requirement of fertilizer. That means managing fertilizer efficiency also. So it gives benefit to the farmers that their inputs cost will be reduced, and the from environment point of view, the climate control point of view, that will be reducing the release of carbon dioxide into the atmosphere. In this aspect, and also the very important thing which hybridization, that normally say hybridization in plants to develop hybrids, but he has hybridized soils also, that bringing natural soils from our worshipping plants like people, religious, and this Bengalensis, but it's very important. The people, thinking is beyond, it's out of box thinking that is being employed, and it's very important for the future. Our future is bright from that point of view, and how we look forward to manage our natural things from that point of view. And also that he has mentioned about the uh, we always talk about now today genomics, that we make use of transcriptomics, metabolomics, proteomics in plants for their further improvement. But here now the soil is also being linked, soil health is being linked with these genomics omics technologies, which is, I think, beyond. I think the new science concept can be developed, that teaching of the new courses from the future point of view, so that people have holistic knowledge rather than we had in the past, and one streamline we used to go, and then we say that I am only soil scientist, I don't know about plants, and I am only plant scientist and I don't know other things. So this is the futurist thinking and out of box thinking. I think Dr. Srivastava has done tremendously well in educating our viewers on all these aspects. And then bioprospecting, soil plant health care through consortium based microbes, which is good relationship between soil constituents, and then agriculture, soil fertility, and human health. So ultimately, how beautifully he has linked the soil health and how to improve it, and then linking with the human health, because all the nutrients we talk about, we always talk about that for human health, our plants are rich in vitamins, minerals, this thing and that thing. So where all those minerals comes from, they come from the soil mood only. So plant may have absorption power more, but they must be available in the soil, then plant can absorb. So those things have been very nicely elaborated and portrayed before the viewers. And the futuristic scientific and societal challenges that he has mentioned, that we need to have complete knowledge of our soil, which is a capital and which is a source where our plant will stand, which we talk about, but we don't talk about the mother of those things which share the weight of plants where it grows on. I think we have had a tremendously well and totally different talk, which opens up the new vistas for the future, for um, a new course development also from the future point of view. That's beautiful. I congratulate Dr. Srivastava tremendously, and I think we too must give a big applause, and viewers must also give big applause to the nice talk that we have. Thank you very much.
Thank you, sir. Thank you so much. Thank you. It's a pleasure talking before all of you. I've thoroughly
Thank you,
enjoyed.
Dr. Kalia. I think we had a fantastic technical talk as Dr. Kalia has elaborated it. I need not to elaborate it further, except saying few words. This your he has clearly brought out, justified the topic. The topic given was the soil health and the human health through crops or fruit crops. Fruit crops because he is working on fruits, particularly on citrus, and which he has clearly shown in his talk that how he has made wonderful, rather very practical and useful contribution to the citrus industry, which will have the effect, not immediate one, but the in the years to come. The fantastic relationship which I anticipated, he has elaborated. Ectophyte, endophytes, and human gut biota. The ectophytes there in the what he had talked and given a nice example also of that people soil taken to the citrus orchard, and the citrus orchard started giving a better result. But there are experiments are required on that how they affect the human gut biota, their efficiency or their output or whatever, so that the human health, required human health, what we call the healthy person, they are met. The origin of the crop on which we depend as a food, and the food leads to the health, whether you call it a medicine or not, which he ended with the saying of a great person that your food is a medicine. That's right. It's a medicine also, medicine in the sense that it keeps you healthy. And so many terms which Dr. Kalia has highlighted, he has brought the new areas of research. Overall, fantastic presentation,
Thank you so much.
and a lot of topics to research upon. He has given several leads. He himself has contributed bringing out the consortia for the citrus on the crop on which he is working, and that is showing the results. We need such type of research, and the organic movement is already on. So organic things, only the organic, when we talk about the soil, you can't keep the organic farming or organic talk separately. In terms of soil, you have to do it. So that is very important. But Dr. Srivastava, science is a science, and we should understand and thoroughly this science for the benefit of the society.
Sure.
I'm wondering, this is a food for thought for everybody and particularly for you. We are emphasizing soil-less farming, and in that also the hydroponics, where your soil is out, but yields are fantastic, nutritionally and quantitative also.
True.
That is there. That is why I said science is a science and we should understand the science. It is not working in one direction, it is working in a multiple direction, in a multiple ways, in a multiple mode. That all that we have to choose depending upon the circumstances and serve the society. That is the one. Your talk, I think, will be referred time and again by the students of not only soil science, horticulture, and the food science and the health experts on that. And the nutriomics, the new area, really needs further research on that. But we should not forget at the same time, I was anticipating that Dr. Kalia will raise the point that in every crop, there is a genetic potential, and we breed the varieties for that character genetic potential to yield better, and not only yield better in quantitatively but qualitatively also. So all those links are to be established. Gene and soil nutrient status, how do they behave? We breed the variety which generally we call, I don't know, my knowledge may not be that deep, fertilizer responsive, nutrient responsive varieties. So all those things are to be worked out, and a simple package is to be given to the farmer and to the people to understand it, not our this complicated thing which we experiment upon in the laboratories. So I think we will be able to contribute better to the human health through plants taking care of soil health. With these words, I thank again Dr. Srivastava, Dr.
Thank you, sir.
Kalia
Thank you, sir.
for their time and the labor Dr. Srivastava has put in preparing a nice talk. Thank you very much.
Thank you.
Okay,
Thank you,
we
sir.
meet next time. The next time we will be meeting on something.
Any questions, sir?
I don't think.
Or appreciations?
Lot many, lot many. So next time they say, can you recommend fruiting plants for the terrace garden? See, such type of things are there. If there is any question worth Dr. Srivastava's status, I would have told it. But they are all just like that. So please excuse Dr. Srivastava. So the next talk will be on something a different crop, what we call a sea buckthorn, with which I am associated, or I was associated, or I will remain associated. Let us meet on 14th, 7 p.m. Thank you so much. Good night, everybody.
Thank you, sir. Thank you, and good night.